Okay, I will show you this video. To understand how DNA is a molecule packed inside the nucleus. So DNA molecule is a large molecule, and some textbook explain us if you give a small cord with 5-10 kilometers long, like 10 kilometers long thread or small nylon cord, and can you pack this inside the tennis ball? Something like that kind of a challenge DNA molecule has. So now we can understand if a nylon cord is 5 kilometers, 10 kilometers long, it's not that easy to pack into a tennis ball. So a tennis ball is a nucleus, DNA molecule is a nylon cord. That kind of a challenge the DNA molecule has. Now, if we look this video, we can understand how DNA packed with the histone proteins to make a condensed chromosome. So this is a DNA molecule. Now let's see what's going to happen. Okay, now these are the histone protein. The blue color one are the histone protein, right? Here you can see histone protein. And wrapped like this. It's coiled to make the nucleosome. So these beads-like things are called nucleosome. First, it just turns two times, roughly 2 times around this histone protein to make the beads-like things called nucleosome. Now they go into a coil again like this. And again, that one going to coil again by wrapping around around it. And again, that structure wrapped around with around it, like a coiling, like a super coiling, right? So then they form these chromosomes inside the cells. Okay, so you now learn if cell is not dividing, just normal cell, that's how the appearance, because some are actively involved in proteins, so they're loosely packed, others are tightly packed, and they don't make proteins. Now when cell is dividing, so chromosome or the DNA duplicate S phase comes something happening like this. Now chromatin is a DNA plus protein, histone protein. Now you can see the chromatin made by the turning around these histone proteins, DNA molecule, and tightly packed, right? So now the first thing is chromatin condense and chromosome visible. Chromatin condense and chromosome visible. That's the first appearance of the prophase, right? But let's see how this happened. We understand like this, right? So if we want to draw a chromosome, we draw chromosome like this. Okay. So this is the appearance of chromosome. then we write, okay, this is the chromosome. And where is the DNA now? DNA is uh, DNA is so DNA molecule is compressed like this to make the chromosomes. So it's compressed to make this chromosome. Now, what is this one? In front of that, we can also find a similar shape. Now, I told you the DNA molecule S-phase duplicate or replicate or synthesis. Okay. If it is make two DNA molecules, it should be like this. It should be like this. But S-phase, when they make two DNA molecules, they don't separate from each other. They copy, but they don't separate. What does it mean? So I can draw it like this then. Here DNA molecule, and the DNA molecule is copied. When it copied, they are not together like this. Not like this, they're like this. So they don't separate from each other. And here, the new DNA molecules, copied one, is the one I can show in white color, this one. It's an exact copy. And again, it compressed with the histone proteins to which that's a visible, right? That means visible mean if they are not compressed, like in previous case, the electron micrograph, they are not compressed, we see only white areas, but heavily compressed areas, we see black. Now here, compressed, and they have now structure. That means they make a specific structure called chromosome. They get together, right? So this is the replicated DNA. Let's say this one is the white one is replicated DNA. And this one is called chromatid. Or it's called sometimes histochromatid, right? Just give me 1 second. Right. Okay, now I think you can understand what is the chromosome structure. Yellow color one is the compressed DNA molecule with histone. What is white one in front of that? It's a copy of this yellow color one. At the end, we can't identify what is original, what is new, because both are same. Both are exactly same because it's exactly the duplicated one. So we cannot see this is the original, this duplicated one. It is just both are exactly the same one, replicated DNA. And the name is given chromatid. Chromatid is the name for given the duplicated one. Replicated one is a chromatid. Okay, now when we look the DNA molecule, the chromosomes, we can see the attachment is mostly in the middle, like an X-shaped. It's in the middle. So how it exactly attached? So it is very tightly attached in the middle region like this, and the attachment to this middle region is called centromere. Centromere is the attachment of two sister chromatids. Centromere is the attachment of two sister chromatids. Okay, then what is the nature of the centromere? It is a region of DNA. Two regions of DNA, like this. It is a one region of like this DNA molecule. The other DNA molecule is like this. So these two regions are attached to each other. In the middle. So that's why we draw like these X-shaped chromosomes. Okay. We are not going to learn detailed structure of the centromere and two DNA molecule, where is this attachment and what protein molecules can link this two DNA molecule, and all these things we are not learning, but we just learn centromere is the attachment of two sister chromatid, right? So now you see chromosome, chromatin, chromatid. So chromatin is the arrangement of the DNA with histone in the nucleus. Chromosome is the X-shaped structure formed after DNA replication. So chromatid is the one sister chromatid of the chromosome. So two names are similar, chromatin, chromosome, and chromatid. But they are structurally they are different. It is very important for understanding to keep what is chromatin, what is chromosome, and what is chromatid, right? Okay, there's another proteins which going to attach each other. Now here, before we proceed to that one, you can see how it looks like when a cell form chromosomes. I mean, we draw like this, and here in the drawing they nicely show X-shaped chromosomes like this. Here one, and here one, and here one, and here one, four. But if you understand such a small nucleus, 46 chromosomes, and even after it duplicated, it's very dense. So it's very dense. We cannot clearly see like this appearance. We can't see like 46 chromosome molecule like this, right? Because the 46 become now 92. 46 chromosome become 92 after duplication, right? So we have to count. This is the one chromosome, and that's the second one now. 46 chromosomes duplicated. So it should be 92 then. But 92 one, they attach to each other like this. It's not separated yet. In a cell, we don't see like this drawing. We can see something like this. Like it's very, here you can see in this picture. Because they are get together and it's like a very close with each other, because we cannot count exactly this 46 duplicated one, right? Okay, then second one, the protein which attach the sister chromatin is called cohesin. Cohesin. So cohesin proteins, it's a protein, right? Cohesin is protein. So cohesin link the DNA sister chromatin with each other at different levels. So they are not very organized in a very special order, but we can find certain point of this DNA molecule. The cohesin proteins attach sister chromatin with each other, and centromere is the middle attachment, and it's a very strong attachment, these two sister chromatins. Okay, so we learn chromosome molecules, and there is a small mistake in the textbook, the text of resource book. Chromosomal arms of sister chromatids attached by special proteins called cohesion. The cohesion is it's like a verb, like a process, like process, cohesion. Cohesin is the protein, so it's a small typing mistake here. It shouldn't be cohesion, it should be cohesin. The protein name is cohesin. Because we have seen water molecule addition and cohesion, so they can make bonds. So making the bonds with this protein is called cohesion, and the proteins are called cohesin, right? Number one, we learn chromosomes visible. Propase chromosomes visible. Number two, nucleolus disappear. So nucleolus is again a dense structure here. So nucleolus we don't see now. It is trying to disappear. Nucleolus disappear. Second one, right? And number three, spindle form. Now, what is spindle? Now it's coming the role of centrosome. I told you a G2 base. This is a centriole. This centriole duplicate. So it make another one like this. Right? So we call centrisome. This structure, this pair together we call centrisome. One is called centriole. Now G2 base duplicated. Okay, if we go back and try to understand what is this structure of centriole, it is made out of this tubulin protein, microtubules, right? Microtubules. So tubulin proteins like this, 13 tubulin proteins joined together to make these centrioles, the microtubules. Then the microtubules organized in three, like this: 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, like this. 9 microtubules, triplet of microtubules, 1, 2, 3, 4, 5, 6, 7, 8, 9, make the centrioles. It's like a collection of microtubules. Now this microtubules formation, and it's a make it long fibers like thing, can be done by the centrosome. So it's like an organizing center, microtubule organizing center. The microtubules, if it is extended, if it is getting longer, and we call spindle. So we call it spindle now. The name is given here, spindle. Spindle is the long extended forms of microtubules. Long extended form is the spindle. And spindle forms not only one direction, it is formed around circle like this. If this is the centrosome, and they form like this. When they form like this, it has like a very special star-like shape, and we call this shape aster. So they're like a star shape because it forms microtubules around it. It forms around it, and only the one they can extend more. So in this picture, you can understand they can extend more towards this direction because space is there, they can extend. But others remain here to the other side. Other side is the cell membrane. So they make a short microtubules. Toward the cell, they make a long microtubules, and they are called spindle. Spindle is the name given to this extended version of microtubules. So they form from both centrosome, and we call here the name given to this called pole, and this is opposite pole, because in a sphere, one end is called the North Pole, other one is called the South Pole, like Earth. Same way, the cell is round shape, and one side is one pole, the other side is another pole. Right? So now here the spindles start to form. That's the third one, right? Third difference is a spindle form. Chromosome visible, nucleolus disappear, spindle form. So three differences we can find. So again, tubulin protein make these microtubules, and microtubules form the spindle. So here you can see spindles. The blue color ones are the long spindle, and they fuse with each other, right? Okay. So you learn what is aster. Aster is the formation of spindle around the centriole. So you get this star shape, like things called aster. Now here is the early mitotic spindle. That means the prophase, they start to make the spindle. Now the prometaphase, end of the prophase. End of the prophase, we can see two changes. Right? Two changes is, one is the nuclear envelope disappear. Fourth one, nuclear envelope disappear. It's breaking its membrane. Nuclear envelope is disappearing. And the fifth one is chromosome attached to kinetochore. This is the centromere. I draw it round shape, right, for the easiness, and that's one chromosome arm. This is the chromatid, second chromatid, like this. Just like this egg-shaped one. Now here is the spindle at the centrosome, and these are the aster. How it see like this? That's a spindle, and this is the aster, and this one is a centromere. This is a chromatid. Two chromatids, sister chromatids, right? Okay, then spindle attached to centromere with another adapter proteins, and that's called kinetochore. So these names are a little bit of difficult sometimes initially to remember. Kinetochore, aster, spindle, right? I told you the remembering the process. First, familiarize with the name, then the order, then what is exactly happening. Like propase, 5 changes. Right? So chromosome attached to kinetochore. That's the last one under pro-metaphase. So we can summarize these events like this. You need to remember only these points. Propase, chromosome begin to condense and visible. That means chromosomes are visible. Nucleolus disappear, spindle form. Pro-metaphase, nuclear envelope disintegrate. Disintegrate is disappear or disintegrate is the same. Chromosome attached to kinetochore. Right? So these are the five things you need to remember. That's very important. After propase, what is the next? Now, if you remember earlier, what is the next is metaphase. So metaphase is the next phase. Now, what will happen in metaphase is like this. Now we can see the arrangement like in this picture. Is everything is now organized in the middle. This is the centrosome, and this is the spindle. So chromosome is attached like this. So you can say attachment here is the centromere, kinetochore, and the spindle. Right? This is the spindle. This is the centrosome. This is the kinetochore, the yellow color one. And this is the chromosomes. Okay. Earlier we have 5 changes. But here, it's easy, you can remember, it is only 1 change. What is the change? Chromosomes organize at equatorial plane. Now, chromosomes organize at equatorial plane or metaphase plane. That means this is the line, middle line. All chromosomes now visible at the middle. That is given us slightly different names like equator, equatorial plane, metaphase plane, like this. Names are given. So all the chromosomes are in the middle, they attach to the spindle. If I take the second one, it's like this. Third one, so we can expect 46 chromosome in a line, right? 46, and this is a one, this is another one. That means 92. So one side 46, other side 46, then 92. Now you understand here, both are same. Both are same mean they're exactly same. Exactly same mean this one and in front of this one, they are same. And this one and what is here in front is same. They're same. So one side has 46, opposite again 46, and they exist in exactly same, the similar, the pairs, duplicated one. So then the two sides are mostly same. If we make a line, middle line like this, and what is chromatid in the other side, chromatid in this side, we take all collectively, they are same because this side has 46, this side has 46. Okay, metaphase, chromosome organized in the equatorial plane. You have only one to remember. So it's a little bit easy, just one to remember. Then the next is anaphase. Anaphase, again one change. What is change? Microtubules spindle. These spindles are getting shorter. Spindles are getting shorter. As a result, they pull one arm of chromosomes. That means the chromatid, one chromatid. Example like this. So this side is full and this side is also full. They try to pull from both poles as it getting shorter. This leads break from the centromere. So it detached from the centromere. Chromatid detached from centromere. Spindle become shorter and chromatid detached from centromere. Just one change to remember. Anaphase 1, metaphase 1. Only prophase 5. Then last one, telophase. Telophase, again one change. One change mean what happened in prophase, opposite is happening. Opposite is happening. What is opposite? Prophase, we said chromosomes visible, condensed. Opposite is prophase. Chromosomes not visible. Nuclear envelope forms. Nucleolus forms. It's disappearing. Prophase, they disappear, and in telophase, they start to appear again. Nuclear envelope, chromosomes not visible, nucleolus start to form. So easy we can remember then, opposite of prophase start to take place in telophase. So I think if you familiarize with the names, I mean just go read again and again these names, telophase, anaphase, like things, If you read many times, you remember, and systematically, like 5 changes happen, one, and opposite of that in prophase. Like this, some kind of a systematic remembering can easily remember this mitosis. Once you familiarize with mitosis, meiosis is again very easy. So when you familiarize with mitosis, meiosis become very easy. So then you don't want too much energy, too much time to remember the mitosis and meiosis. Okay. Do you have any questions about mitosis? Anything to ask? No? Okay, next is meiosis, right? Okay, you don't have, then we proceed to the meiosis. I think if you have a good idea about mitosis, I am only focused on the important thing in meiosis. Otherwise, most of the things same as mitosis. Now, significance of meiosis. Significance of meiosis, right? As a very important thing. It's very important things, and we can't learn everything in this lesson because we have to learn few lessons in the future to understand some of the concept. One thing is for sure like this. If cells are produced to make the organism, like I discussed before, ovum, and this is the sperm cell. And this being 23, and this being 23. And this may be the first child. And let's take the same family, again ovum and the sperm fused together, which being 23, 23. And that's a second child. We know that they are not genetically same. They are not genetically same. So I'm talking about within a family. If we take families, the different families, they are anyway different. Now, how we can understand if a mother has same genetic information and father has same genetic information, that's a mother and it's a father. And why the children are different in the family? Because father is providing his genetic information, mother is providing his genetic information. Now, the rule is like this: 50% information is coming from mother, and 50% information coming from father. Then half mother's information, half father's. Why 50? Because total is 46. 23 from mother, that is 50%.